everyone. It's my honor to be here to show you some of my research data today. My topic is using biomarkers to understand past climate. What can lakes tell us? The two keywords here are biomarkers and lakes. Before I jump into the research, please allow me to introduce myself a little bit. I started my undergrad study in China University of Geosciences in 2011. This is where my university is. In my junior year 2013, I got some research grant, and I was lucky enough to be able to go to the state key laboratory of biogeology and environmental geology to do some independent research on biomarkers. That's the building I always considered that the birthplace of my research interest. I also went to the neighbor university called Huazhong University of Science and Technology, study English translation. So I received my two bachelor's degree in 2015 June. After graduating, my project is close to be finished, but not yet. So I went to the desert in Xinjiang, where is the northwest China, to have some soil sample collected and add those data into my work. So by the end of 2015, I was able to wrap up my work and get the paper published. After that, I was an active performing pianist and I taught piano. I was a part-time translator for the expo and traveled a lot. I went to Tibet, Vietnam, and Thailand. So I was really considering what I want to do for my life. During my trip to US, I had a chance to meet with my current advisor, Dr. Joseph Werner. And he was very kind and gave me a lab tour, introduced me to all this machine he had in the lab. And he told me that they went to Lake Chalco in Mexico and had a super long sediment core recovered from Lake Chalco in Mexico. And he's looking for students to join this project for biomarker analysis. After hearing that, I was super motivated. So I applied for a PhD with him. So lucky enough, I was accepted and started my undergraduate study here in Pitt in 2016. So while hearing that Chalco core in Mexico made me so excited, let's answer the first question. It's like, what can lakes tell us? Because sediment is transported by wind, water, ice, and less commonly biological agents, and they can preserve sediment cores can preserve various proxies for past climate and environment, such as pollen, like the picture here, and some plant remains, and molecular fossil, algae, charcoal, and planktic and benthic organisms. While our instrumental measurement of past climate change of climate variables are only available over the past century or less, lake sediment can go beyond the instrumental limit. Unlike tree rings and ice cores, lake sediment are less limited by regions. And why do I care about South and North America and why specifically Chalco made me excited? Because there are around 175 million population in this area here. It's one of the mostly dense populated area in the world. Mexico and Central America are at risk from the impact of climate change in the coming decades because they will be exposed to greater range of climate changes and have a relatively weak adaptive capacity when compared to the world at large. Within the region, climate change is evident in increased temperature, changes in precipitation, and sea level rise. Its intense shoreline and this relatively small continent area make this region very susceptible to natural disasters associated with climate change. So what is this charcoal core? Why it is important? In February and March 2016, a 350 meter lacustrine sedimentary core were recovered from this area highlighted here. Chalco is in the basin of Mexico, and this site has a high potential to be the optimum position to elucidate North American tropic climate change, and that will enhance enhance our understanding of the climate mechanism in this mid-latitude to tropical area in the continent. Mexico and Central America are projected to face drier climate in the future, which will exacerbate their hydrological stress. And understanding this past climate change and the climate mechanism in this area will help their people better prepare and adapt for future climate change. It will also help their government to make policies dealing with climate change, thus to maintain their social stability. So how to analyze this core? The tool I'm using is called Biomarker. Essentially, biomarkers are complex carbon-based molecules derived from formerly living organisms. They can be characterized by their structures and isotopic content. It will also indicate the biosynthetic origins and environmental conditions that those living organisms have experienced. And using biomarker, I'm going to develop calibration. So calibration is a very abstract conception. In my research here, it means a way of checking and modifying the accuracy of biomarker-related measurement or observation. So the whole thought process will be that we extract biomarkers, measure their abundance, distribution, and their isotopic content, and we use statistic models to help to turn those measurements into something more directly comprehensible, like temperature and precipitation. So why specifically precipitation and temperature? Because the simplest way to describe climate is to look at average temperature and precipitation over time. So here's the structure of my work. To look into precipitation, I'm going to use the biomarker called hydrogen isotope values of terrestrial leaf wax. So I'll call it delta D wax. And for temperature, the biomarkers I'm going to use is called bacterial membrane lipid-derived brGDGTS. And with these two biomarkers, I'm going to have some statistic models to help to develop the calibrations for South and North America. And it will be the regional specific calibration to help us to reconstruct precipitation and temperature. So let's take a look at the first part, using delta D wax to reconstruct precipitation. So there are two stable isotopes of hydrogen. One is protium and the other is deuterium. And the difference is that in deuterium, it contains 1 neurons. All water molecules consist of 2 hydrogen and 1 oxygen. When one ordinary hydrogen atom replaced by deuterium atom, it will be heavier. So HDO molecules are heavier than the standard H2O molecule. The most fundamental description of hydrogen isotopes in a system is the relative abundance of deuterium and protium, or ratio between deuterium and protium, denoted as R here. So this is how delta D is calculated. It's actually very linear. To make it simple, the more deuterium it has in the sample, the higher delta D value it will have. So let's take a look in leaf wax. Leaf wax is a cuticular layer on external surface of a higher plant that could help them maintain water balance and protect the plants from abrasion. Leaf wax normally comprise linear very long chain compounds, including varying ratio of acids, primary and secondary alcohol, esters, and alkanes. Hydrogen isotopic value of leaf wax, which is delta D wax, has been widely used to reconstruct paleohydrological change in the tropical area, where meteoric water source, rainfall amount, and vapor transport pathways are primary control on the hydrogen isotopic composition. So this is the mechanism chart here. Blue axis shows the direction of deuterium enrichment, or higher value of delta D. Evaporation and transpiration could happen when water is preserved in soils and leaves. And in both processes here, lighter hydrogen isotope will be more preferred, leading to the enrichment of deuterium. When enzymes using leaf water to form leaf wax lipid, they will also prefer using lighter isotope. So this will result in a depletion of deuterium enrichment in leaf wax. And net fractionation, or sometimes people call it apparent fractionation, are used to describe the difference between the initial product, which is the rainwater, to the end product, which is the leaf wax lipid. So to sum it up, there are three processes that are likely to affect the delta D wax value. First is the isotopic composition of meteoric water, which is the resource. And then the second is the deuterium enrichment of soil or leaf water due to evapotranspiration. The third will be different plant groups because they will have different enzymes that will affect the deuterium value in leaf wax lipid. So my research question here is, can leaf wax delta D record delta D water signal, therefore be used in reconstructing precipitation? Because previous studies showed that in southeastern Mexico and Central America, delta D wax is independent of the isotopic composition of precipitation, and indeed is largely controlled by aridity index, which is defined as mean annual precipitation over mean annual potential evapotranspiration. And the other environmental factor that will control the delta D wax value will be vegetation type. So my research hypothesis here will be, despite the sophisticated environmental process affecting the delta D wax value, in this region, when we have less vegetation change, leaf wax delta D is still mainly influenced by precipitation and meteoric water delta D. So this is the sample site map. We actually collected over 80 lake surface segment sample, but we couldn't retrieve most of the delta D wax measurement in them. So that's why in this map we have less samples showed here. And because previous research revealed that in Yucatan and Central America, which is denoted as YCA here, wax did not necessarily record water, so I divided this research area into two subregions. One is this YCA region, the other one is the Mexican Plateau area, denoted as MP here. And there's other reasons why I divide this study area into two regions. First is the topography, because in Mexico and Central America, it's generally a higher altitude area. The average altitude in the sample site is above 2,000 meters, while in Yucatan and Central America, it's generally lower altitude. The second region is climate zones. The climate zones in Mexican Plateau is more complicated compared to that in Yucatan and Central America. Due to the lack of weather stations to collect actual meteoric water samples, in this study, there is one very important assumption. We assume lake surface water hydrogen composition, which is delta D water, are similar to that of the meteoric water. We acknowledge the study area have drastic seasonal contrast of precipitation and evaporation, which will affect the delta D water signal. So the majority of the sample were actually collected during wet season to minimize the effect of the intense evaporation, so we can reduce the potential bias in ionized delta D water. We realize that it's not perfect, but that's the best we can do here. Let's take a look at the data here. So in this figure, the left panel is the data from Mexican Plateau, and the right panel in the yellow box are data from Yucatan and Central America. So first, let's see the delta D water. The range of delta D water in Mexican Plateau is around 61 per mil, and the delta D water in Yucatan and Central America is 66 mil. They're very close together, but there is statistic difference between these two sample sets, which means that they will have different source of rainwater, which is highlighted in both charts here. And next, let's take a look at the end product, which is the leaf wax lipid delta D wax here. In Mexican Plateau, the range of delta D wax is around 66 mil, and in Yucatan and Central America, the range of the leaf wax delta D is 37 mil. Compare the range from the source, which is rainwater, and the product, which is leaf wax lipid. We can see that leaf wax can not fully explain by the delta D water here, especially in Yucatan and Central America. And compare these two data sets here. In Mexican Plateau and YCA, there's no statistic difference between delta D wax in Mexican Plateau and YCA. So to answer the question, can leaf wax delta D record meteoric water delta D signal, therefore be used in reconstructing precipitation? Let's take a look at the data here. So the left one is data from Mexican Plateau, and the right one in blue dot is the data from Yucatan and Central America. The x-axis is the distribution of delta D wax, and the y-axis are delta D water. Indeed, delta D water and delta D wax show no relationship across Yucatan and Central America. This result is in agreement with another study in the same area that I mentioned before, which demonstrated that aridity index and vegetation composition are the two major environmental controls of delta D wax. On the contrary, in Mexican Plateau, delta D water and delta D wax actually show a strong correlation with R squared equals to 0.7. And when we compare apparent fractionation, which is used to characterize the hydrogen isotopic fractionation between source and product, which is water and wax. It will represent the sum of many individual fractions effect on both physical and biochemical process. And we can see from here this blue arrows is using to describe the apparent fractionation, and they are different in these two regions. And an important assumption in the application of delta D wax as a proxy for reconstructing delta D of precipitation is that the apparent fractionation does not vary too much. And we can see from here the left panel, which is Mexican Plateau, there is a linear relationship between delta D wax and delta D water, but there's no relationship between between these two in Yucatan and Central America. So this further approved that the net approximation are more constant across Mexican Plateau. Thus, delta D wax are more promising to be used as proxy for reconstructing delta D water in this region, Mexican Plateau, than in Yucatan and Central America. So what's the reason that this delta D wax and delta D water are pretty linearly related in one region but not the other? Is it because aridity that was suggested to be the primary control in YCA area? So we take a look at the meteoric water line here. For those of you who are not familiar with meteoric water line, I will explain a little bit. So the black solid line here is called global meteoric water line. It's used as a reference. And we have two lines here. One is the orange line, one is the red line. The orange one is from Yucatan and Central America. It's the local meteoric water line. And the red line is used to sample from Mexican Plateau to draw. It's the Mexican Plateau local meteoric water line. So we can use this angle between the local meteoric water line to the global meteoric water line to roughly get an idea of the aridity in the two regions. So the bigger the angle, the more arid the region is. So when we compare these two local meteoric water lines, we realized that they have similar angle with the global meteoric waterline, which suggests that these two regions are probably having the same aridity. And also we can see here the x-axis is delta dx and the y-axis is aridity. For the data here, we did not see any relationship between delta dx and aridity. So aridity might not be the main explanation here why these two regions are different. So if it were not aridity, can it be vegetation cover? So here is the vegetation distribution map in my study area. And we can see that in Mexican Plateau, all my samples are collected in the tropical and subtropical coniferous forest, which is like this very light yellow color here. While in Yucatan and Central America, our samples were collected in different vegetation sites. So that could be it. That could be possibly due to different vegetation cover that caused the difference of these two subregions, but we do need further evidence and analysis to address this question. So my conclusion here for the first chapter will be that LeafWax delta D can be used to reconstruct regional delta D of precipitation during wet season in Mexican Plateau, but may not necessarily be a good proxy for the same purpose in Yucatan and Central America. Alright, now let's take a look at using bacterial membrane lipid called brGDGT to reconstruct temperature. What's the calibration look like here? So we started, what is GDGT? Full name of GDGT is called branched glycerol dialkyl glycerol tetraethers, and that's where the abbreviation comes from. We generally just call it branched GDGTs. They are membrane-spanning lipids. They are widely discovered in all kinds of environments. Here's the structure of branched GDGT here. So take a look at their names. So the 1A, 1B, 1C group. So they actually, compared to the rest of the group, they will have less of this methyl, or we call it branch, on their chain. So compare one group, 1A, 1B, 1C, to the two group, which have 2-1, 2B, 2A, 2B, 2C. The difference is that the two group has one more methyl group here. So similarly, the 3 group will have one more methyl group compared to 2 group, and has 2 more compared to 1 group. So the 1, 2, and 3 are used to differentiate the number of methyl or branch on the main alkyl bond. And when we compare A group with B group will have one ring, or you can call cyclopentane moieties, on the chain, while C group will have 2 rings of cyclopentane moieties on the chain. So A, B, and C are used to denote the different numbers of the rings on the chain. And there's difference between 5-methyl GDGT and 6-methyl GDGT isomers. Their chemical formula is the same. However, the only difference is where this methyl group is attached to the chain. In the 5-methyl, let's say the 2 group here, the 5-methyl branched GDGT has this branch attached to the 5th carbon on the chain, while the 6-methyl isomers have the branch group attached to the 6th carbon on the chain. So that's the difference between the 5 and 6-methyl GDGTs. So that's everything about the structural difference. Take a look at why we think they're important. Because over the last 2 decades, They're ubiquitously found in soils, peat, marine lake sediments, and water column. They are very chemically stable, so after they are preserved in the sediment, they're not going to degrade easily, or their chemical structures are not going to be altered easily. They're also relatively easy to extract in the lab, and they are very sensitive to the ambient environmental change. So they have become a very important research focus for organic geochemists because of their application in temperature and pH reconstructions. Basically, they are used to reconstruct mean annual air temperature. They can also be used to reconstruct water temperature in the lake, in sea, and they can also be used to reconstruct pH. Basically, when we talk about the structural change, they have different methyl group and the ring group. This bacteria will actively adapt to environmental change by just altering the structures, and that I'll explain later. And the potential problem of using GDGTs as a temperature proxy is that they might not just respond to one specific environmental variable. Let's say they can be responding to both pH and temperature, and it is very hard to isolate a single environmental factors out. And second is that we don't know who produced them. We know that there are bacteria derived membrane lipid, but which bacteria we don't know yet, and there's no successful lab culture so far to identify their producer. The third problem is, are they producing where they're found? If they were not, then they might not necessarily record the temperature or pH in that specific area, because if they were transported from further somewhere, then they might record the other place's temperature and pH. And the last problem is regional difference. And that's the whole reason why we need a regional calibration for South and North America here, which I will also explain later. So my research question for this part is, can we use branched GDGTs to reconstruct temperature in my study area? And my hypothesis here is yes. I believe that in this area, branched GDGTs still mainly respond to temperature change and has minimum influence from GDGT, from pH or other environmental factors. The second research question is, how can we improve the precision of our calibration? And my hypothesis here is that by developing regional specific calibration, we will be able to reduce biases in temperature reconstruction. So this is the sample map here. Actually, we were able to extract GDGTs from most of our sample collected compared to that of delta DWAX. So that's a good sign. And we have a co-inertia analysis based on the Euclidean model, and we try to summarize the relation between variations in samples and to see that if there's any regional difference of these branched GDGTs. And as we can see here, we do not find any obvious 2 or 3 or more clusters of samples. So I do not divide the entire region into 2 subregions like what I did in the leaf wax study. And here, the purple or pink color is the sample from my research area, and the blues are the samples from African lakes. And just by comparing these two, we can see that all GDGT has different distribution between these 2 sample sites. When we take at the ternary plot here, we also notice that so the yellow triangle are the samples from Chinese lakes, and the purple ones here are African lake samples, and blue ones are my sample from Mexico and Central America. And we can see that they fall into different clusters, which means that these 3 different regions have like different GDGT distributions. And that's why having a regional calibration is important and will potentially improve our precipitation. So talking about calibration, there are two major types of them. One is called proxy-based calibration. The other one is called fractional abundance-based calibration. So let's start with the proxy-based calibration. There are two important indices calculated. One is called degree of methylation, denoted as the MBT, and is calculated as the one group over the total abundance of all other GDGTs. And CBT here is called degree of cyclization. So if you remember that between A, B, and C group, A group has no rings or cyclopentane, and B group has one rings, and C group has two rings. So degree of cyclization is used to describe it, and it's calculated as the negative log of 1B plus 2B over 1A and 2A. So it's the group with one ring over the group of no rings, and with the log calculation here. So MBT is widely known to be the main proxies for temperature, and CBT, you might notice the log here, are mainly the index for pH. And here we have the similar indices, but this time we exclude all the 6 methyl isomers. We just use the 5 methyl isomers and perform the same calculation for the MBT and CBT index here. The reason to separate 6 methyl isomers are some research has demonstrated that 6 methyl isomers are mainly respond to pH or alkalinity. They are not necessarily respond to temperature. The second reason is that if you incorporate them into the calibration, considering that they might not be responding to temperature, they will introduce scatters in your calibration. So that's why sometimes people will separate the isomers. Let's take a look at what the calibrations look like. So there are examples of like 3 different calibrations. First one is the study in China, and their calibration include both MBT, CBT, and the second one was used to reconstruct the water temperature. It's a study in Greenland, and it includes the calibration, includes only the 5 methyl MBT. Similarly, the study in Africa, where the researchers use the branched GDGTs to reconstruct the mean annual air temperature, they have only one index, which is the 5 methyl MBT in the calibration. So the question here is like, we already have so many different proxies-based calibration. Why do I still have to develop my own? Can I just not use them? Maybe they work well. So I explained a little bit of the regional difference of GDGT distribution there. And when we actually try to apply those calibration here, we can see there are bias. So here's the plot that we apply the Chinese sample-based calibration into our own samples. The x-axis is the reconstructed temperature using the calibration here, and the y-axis is the real temperature, and this dashed line is a one-to-one reference line. So we can see that the reconstructed line, which is the highlight yellow one, the regression line here, is pretty far from the reference line. So the closer it got to the reference line, the better the calibration works in general. And we can see that both the calibration based on the Chinese samples and the calibration based on the African lake samples, neither of them works very well in the samples we collected, which also means that in the region of Mexico and Central America. So can we develop our own calibration? And if we can develop our own calibration, does our own calibration work better? So here's the answer. This is what our calibration look like. We incorporate both MBT and CBT into the calibration with the RMSE root mean squared error to 2.5 degrees Celsius with the R squared close to 0.6. And as we can see, the y-axis is the actual temperature and the x-axis is the reconstructed temperature. Our regression line with this calibration is actually overlapping with the one-to-one reference line, which means the calibrations are not very biased. And what about that if we take the 6-methyl isomers off? Because as I mentioned before, the previous research has demonstrated that the 6-methyl isomers are not responding to temperature very well. And we just use the 5-methyl isomers, which I highlighted here. And our calibrations look like this. And this time with 6-methyl isomers being excluded, we're able to improve our R squared up to 0.7 and then reduce our RMSE down to 2.2. So the conclusion for proxy-based calibration here is that 5-methyl branched GDGTS isomers are the main groups respond to temperature, while the 6-methyl branched GDGTs are likely to reflect other environmental variables, such as pH. And highlighted here, this is the recommended proxy-based calibration to use in the Southern North American region with the RMSE equals to 2.24. So what about the other format of calibration, which is called a fractional abundance-based calibration? Here's what fractional abundance are calculated. For example, for fractional abundance of GDGT1A, it's calculated by 1A over the total abundance of GDGT. So it's the same for 2B and 2C and 3C, etc. And for the fractional abundance-based calibration, we're actually trying to find the best subset of branched GDGTs to calculate for temperature. This is a quick look at what fractional abundance-based calibrations look like. The first calibration here are using the samples from East African lakes. And in that region, the best subset samples include 3A, 2A, 2C, and 1B. But in a global soil sample set, to develop a calibration to reconstruct mean annual air temperature, the best subset samples include 1A, 1B, 1C, and 2A. So it's different from the East African regional ones. So we now know that we actually expect to have a different subset of sample of GDGTs for our study region here. And knowing that we're going to perform stepwise regression to find the best subset sample, in order to choose a set of predictors that have the best relationship with the dependent variables, we perform the forward and backward selection. The difference is that the forward selection method, you begin with no candidate variables in the model. At each step, select the candidate variable that increases the R squared the most until none of the remaining variables are significant. And the backward is just the opposite. We begin with the model include all candidate variables, and then we took one out each step until no significant variables remain. So take a look at the stepwise forward selection here. This is what our calibration looks like, and the best subset include 1A, 1B, 2A, and 3A, and all of them are from the 5-methyl GDGT group. And the R squared equal to 0.7 with the RMSE equal to 2.3. And we can see that the regression line is overlapping with the reference line again. It works pretty well. What about the backward selection? Will it be better than the forward selection? So this is what backward selection gives us. The best subset is different with 2A, 3A, and 6-methyl 3A prime. So prime means 6-methyl GDGTs. So 2 5-methyl GDGT with 0.6 methyl GDGT in this calibration. And you'll probably notice that yes, we do have a better R-square here with a similar MSE. However, when you look at the plot here, we notice that the regression line is actually pretty far from the reference line. This is easy to understand because the backward selection, it works its way down instead of up. You're always retaining a large value of R-squared. The problem is that the models selected by this procedure may include variables that are not really necessary, which in another term we say overfitting. So compare these two, we agree that the step forward selection calibration performs better than the backward selection, and the best subset sample comprise all 5-methyl branched GDGTs, and it looks like this. So this is the calibration we suggest to use in the South North America region. And this is a brief look of what the charcoal core looks like. And the next step, I'm going to apply the calibration from branched GDGTs and leaf wax, try to reconstruct the precipitation and temperature in this region using this charcoal core. I'm still doing it right now, so I'm not going to share data today. However, a lot of data already produced by other collaborators. They've already published papers. If you're interested, you can go do a brief search. And I wish that I can share it as soon as possible. That's the end of my topic today, and I would like to acknowledge the support and encouragement I received during this whole research, and thanks Brad for organizing the whole talk. Thank you very much for coming.